Okay, so we're continuing on here where we're going to talk about actually making stuff. So I think the easiest place to begin talking about biosynthesis of macromolecules is actually talking about carbohydrates, because we've spent a considerable amount of time talking about essentially glycolysis, and I'm going to introduce another pathway, the pentose phosphate pathway, that can be used for carbon oxidation, but it's more important for essentially building and biosynthesizing larger carbon molecules from smaller bits of carbon that we'll see in a second. So I think the best place to begin is to go back and look at the thermodynamics of glycolysis. But before we do, we have to go back and think about carbohydrates. And so you need to produce glucose and other saccharides from other smaller oxidized metabolites. And so this is especially true if microorganisms are growing on things like amino acids, for example, that we just talked about. So those things are, 3, 4, maybe 5 carbon compounds, and they are certainly not sugars. So if we want to make glucose, we'll use a process called gluconeogenesis. So I hope many of you have seen this before in Biology 1831, but if you haven't, I'll try and make things as clear as I can. Now there's some key items in gluconeogenesis, and essentially what's nice about gluconeogenesis is it's more or less glycolysis, except for 3 reactions. One is that you need oxaloacetate to be converted into phosphoenol pyruvate. You need fructose 1,6-bisphosphate to be converted to fructose 6-phosphate, and you need to be able to convert glucose 6-phosphate to glucose. And there are other important saccharides as well, 5 carbon sugars, the most important of which being ribose, because you need to have a ribose sugar backbone to make DNA and RNA, and that this relies on the pentose phosphate pathway. And so I will enumerate and elaborate on what all of these things are. So for gluconeogenesis, again, I think it's best to begin looking back at the thermodynamics of the process. So glycolysis, you'll remember, consisted of 10 reactions, and under standard conditions, some of the reactions were very thermodynamically favorable, and many were thermodynamically unfavorable. And you can work out the standard free energy of these reactions and see this. Now the thing is in the cell, this is not under standard conditions. It's not equal molar amounts of reactants and products. In fact, these things are at a different equilibrium. And the equilibrium within the cell actually favors energetically certain reactions down here in red that are very, very energy liberating reactions. Now the problem here, first we'll start with a benefit, is that under equilibrium conditions, many of the reactions of glycolysis are essentially net energetically neutral. Their delta G is around 0. Now if we want to run this pathway backwards, all of these reactions essentially will run forward or backwards relatively easily. Now the trick with glycolysis or gluconeogenesis is that we have to overcome these free energy barriers to producing glucose from, say, pyruvate. So if we want to do that, let's take a look at how that gets done. So shown here is a metabolic pathway leading from pyruvate all the way to glucose, and showing us all of the important reactions that are going to take us there. So we'll note that normally, again, thermodynamically, most all of these stage 2 reactions, all the way down to PEP, are essentially reversible, and these are able to run just in the reverse direction. And remember our aldolase reaction that was normally very, very energetically unfavorable and was only made favorable by keeping glyceraldehyde 3-phosphate concentrations very low. Now this reaction gets to have lots of product and essentially favors production of fructose 1,6-bisphosphate. So if we started from phosphoenol pyruvate, without any effort really, we could make fructose 1,6-bisphosphate. However, that's not what's going on in the cell, right? So you're going to start oftentimes from pyruvate if we're eating amino acids, and we need to make larger carbohydrate molecules like glucose. So say if we wanted to make LPS for E. coli, we need to be able to make glucose. Or say we wanted to make the carbohydrate backbone that is in peptidoglycan. So if we want to make cell wall and we're eating amino acids, you have to be able to run this pathway backwards and build larger carbon compounds. So in order for this to happen, it requires some energy investment. So just like the oxidative direction of glycolysis, we had to have energy investment to get glucose prepared to be torn apart and oxidized. We have the same thermodynamic barriers to this happening for gluconeogenesis. Now what ends up happening is that pyruvate needs to be, essentially, you have to add a CO2 to pyruvate, and this requires energy in the form of ATP. This produces oxaloacetate. Now remember, oxaloacetate is a component of the citric acid cycle. So essentially, we're beginning to run many of the reactions that we talked about backwards. So you need to basically put pyruvate into the citric acid cycle, more or less backwards, to then take it back out again through another energy-intensive carboxykinase reaction that releases that same CO2, and in the process, you've produced phosphoenolpyruvate. Okay? So essentially, you have one investment of energy, a second investment of energy to produce PEP. So all of these other reactions are able to run more or less at equilibrium completely backwards, very favorable. Now we run into fructose 1,6-bisphosphate. So there are dedicated enzymes now that are going to use, that are hydrolases, that are going to use water to remove the phosphates off of fructose 1,6-bisphosphate to produce fructose 6-phosphate. Fructose 6-phosphate can be isomerized into glucose 6-phosphate, and there's a glucose 6-phosphate phosphatase that is a hydrolase that's going to hydrolyze off this phosphate group to produce glucose. So essentially, you have some dedicated reactions to overcome thermodynamic barriers to convert pyruvate to phosphoenolpyruvate. Because remember, this was one of the substrate level phosphorylation reactions that can happen in the oxidative direction. But then in the reductive direction, or the anabolic direction, gluconeogenesis, we have to have separate reactions that make energy investments so that we can overcome the thermodynamic barrier between pyruvate and phosphoenolpyruvate. Okay, so let's dissect these reactions a little bit. And so if we look at pyruvate here, that this pyruvate carboxylase, what this enzyme is doing is adding bicarbonate to pyruvate to produce oxaloacetate, and in the process, this uses ATP. So more or less, this is a ligase enzyme, so EC6, right? So now we're going to make another energy investment, and this phosphoenolpyruvate carboxykinase is going to essentially transfer a phosphate group onto oxaloacetate at the expense of basically liberating this CO2 that was basically stuck on in this first reaction. So through this 3-carbon, 4-carbon, 3-carbon reaction setup, we can get past the thermodynamic barrier that is essentially, moving backwards from pyruvate to phosphoenolpyruvate. Similarly, we can make fructose bisphosphate into fructose 6-phosphate, glucose 6-phosphate into glucose. So both reactions use the same mechanism but different enzymes. So I'm showing here glucose 6-phosphatase that is a hydrolase. It's hydrolyzing off this phosphate group to produce glucose. And remember that this step needs to be coupled to ATP hydrolysis to become exergonic in the basically the oxidative direction. But essentially this reverse direction is actually fairly energetically favorable. So this reaction is exergonic already. The enzymes are actually inhibited by AMP, and the enzymes are activated by citric acid. So essentially, what this does is basically, there are feedbacks on basically how this enzyme responds to the nutritional state of the cell. So if things are good, you want to begin building compounds, right? So if there's a lot of citric acid, it says to the cell there's a glut of carbon, and we need to build stuff, we need to make things, we want to store carbon, or we want to make new cells. Now, if there's AMP in the cell, that's a signal for starvation. So that means that not only did you use ATP to convert to ADP, but things got so bad that you turned ADP into AMP to basically get as much energy out of like ATP as you possibly could. And if AMP is at abundant concentrations, essentially you want to stop making biomass. You want to focus on generating energy and running these pathways like glycolysis in the oxidative direction to generate energy. So, if we want to make glucose from pyruvate, essentially these reactions are all happening simultaneously, and that's the thing to keep in mind, is that you're running things in the oxidative direction, and you're also simultaneously making higher order carbon molecules. So basically the cell is always essentially oxidizing carbon to generate energy, and also simultaneously building more complicated molecules to essentially grow and divide. That we're going to talk about the pieces of this just in a few slides. So ultimately this costs the cell 3 ATP. So we're just going to say functionally that GTP is more or less ATP, and so 3 ATP. And so this is a significant challenge when you're fermenting glucose, but it's not impossible. So there are other energetically favorable things that can happen during fermentation. So you might oxidize some glucose completely. You might stop some glucose at fructose 6-phosphate, for instance, and put that into the pentose phosphate pathway. Yada yada yada. That's beyond where I want to take you in this course. But know that the energetics and accounting of this working does work if you balance reaction usage and oxidative directions and reductive directions to essentially balance the costs of building things versus the energy you get by oxidizing things. So the last pathway that you need to be able to see, you need to know that it exists to understand how bacteria like E. coli can grow on fatty acids and produce nucleotides and everything else that they need to produce from one source of reduced carbon. Critical to getting this done, we saw gluconeogenesis, so we can build hexoses and from simpler carbon compounds like pyruvate, or any other carbon compounds that can enter into glycolysis. They can also be used as gluconeogenic substrates. So the pentose phosphate pathway, on the other hand, is a little bit different, and it's a little bit tricky. And so it's an alternative stage 1 glycolytic pathway, and then it can operate in the oxidative direction. It often operates simultaneously with glycolysis, and the reason is that it generates NADPH instead of NADH. And so this becomes important when we're going to start talking about building things like fatty acids, for example. That you want to make things, make many different forms of reduced carbon molecules. You need NADPH, and so the benefit here is when this pathway is operating, there are 5 carbon and 4 carbon sugars present in the cell, and these are critically important for making nucleotides and aromatic amino acids and other amino acids. So the outputs of this pathway is it produces fructose 6-phosphate and 3-phosphoglyceraldehyde, And so essentially, you run this pathway in one direction, it spits out pieces that are ultimately going to end up in glycolysis anyway, but in the process produces these key intermediates to make amino acids and nucleotides. So let's take a look at this pathway. So what ends up happening in the oxidative direction is you would produce glucose 6-phosphate from PTS transport or hexokinase. And if this is operating in the oxidative direction, that you'll produce 6-phosphogluconolactone, that ultimately this will turn into 6-phosphogluconate, and we've seen this compound before when we saw the Entner-Duderoff pathway, for example. And similar to the Entner-Duderoff pathway, but slightly different, is 6-phosphogluconate dehydrogenase. And this enzyme produces NADPH, and in the process oxidizes one carbon, this carbon here, off of this compound to produce ribose 5-phosphate. Now ribulose 5-phosphate. So ribulose 5-phosphate can be isomerized into ribose 5-phosphate, and ultimately this can go on and be used in making the backbone of DNA and RNA, right? So this can operate in the way that the rest of this pathway operates. It's beyond the scope of this class, but I like you to see it, is that there are a number of transketolase reactions and transaldolase reactions that happen and essentially exchange carbons between 5-carbon, 2 5-carbon sugars, xylulose 5-phosphate, ribose 5-phosphate, and this is one of the strange times in the cell that you see said heptulose, a 7-carbon sugar, phosphate. And actually, in producing LPS, this compound, this 7-carbon sugar, ends up in a lot of LPS. So again, when you're making big carbonaceous things, the cell needs to make these things for lots of purposes. And you have glyceraldehyde 3-phosphate. And so another exchange reaction happens again, and during this process, you end up with erythrose 4-phosphate, and this transketolase will also spit out glyceraldehyde 3-phosphate. And so you end up with this balance of fructose 6-phosphate and glyceraldehyde 3-phosphate, and this is a complex reaction network that, in order to understand the stoichiometry of it, you need to be working under essentially operating under steady state conditions. And essentially you have 10 carbons, and what ends up happening at one point is that 3 carbons go away, you're left with 7 carbons, and then you do another 4-carbon, 3-carbon trade. And so ultimately, in the end, there are the stoichiometry of all these reactions works out, but what's important is you end up with 4-carbon intermediates and 5-carbon intermediates that are critically important for making nucleotides and amino acids. And when operating in the oxidative direction, you produce NADPH via this pathway. But importantly, during gluconeogenesis, this pathway can operate just as well backwards as it does forwards. Okay? So if we're moving from pyruvate and we need to make ribulose 5-phosphate to make the carbohydrate backbone for DNA and RNA, essentially this pathway will run backwards and produce those intermediates. So if we're wanting to make carbohydrates, the biosynthetic pathways we need are gluconeogenesis to produce 6-carbon sugars, and the pentose phosphate pathway to produce 4 and 5-carbon sugars.